Thank you very much for inviting me to talk here, and thank you all for coming. I hope I'm not going to be too boring. I'll try to be as exciting as I can. I'm going to talk about what I actually think is a fairly exciting technique, which it's called STED microscopy, stimulation emission depletion microscopy. It makes your confocal pictures from blobby, kind of ugly stuff into really single molecular, single organelle, two-dimensional pictures. So it's actually one of the, I would say, in imaging optics, let's say, let's not say just general optics, let's say in imaging, in microscopy, fluorescence stuff optics is probably one of the best discoveries in the last 2-3 decades. A couple of little, how should I say, a little Entschuldigungen before I move into the talk. My computer didn't work, so I'm going to have to use one of the local computers here. So if there's any technical problem, I will blame shamelessly the computer here rather than my own that didn't work. That's number one. Number two is it's for the first time I'm being actually filmed when I'm talking. So I have been warned not to curse anymore. I do it every once in a while. I'm not going to do it today because it's probably going to be on tape. And I'm going to be blackmailed with it for the rest of my life. So I might be a little less entertaining than usual because of that. So without further ado, I think I will just move into the talk. I will kind of stay in this corner. I have a mouse here with which I can kind of move from slide to slide. And I guess what interests you is on this side rather than my shadow here. So let's just jump in. So what we actually work on are synaptic vesicles. This is an electromicrograph section through a frog neuromuscular junction. On this scale, the terminal would be much larger than the whole institute. They are really long. They are a few hundreds of microns long. But what in section, in a longitudinal section, in a transversal section, what they look like is the typical synaptic view. You have a clump of synaptic vesicles. You have a darkening of the plasma membrane at what is called the active zone. This is where the vesicles are going to fuse and release a neurotransmitter. Perhaps this little bump here is just a vesicle that just has fused and then its membrane may be diffused a little bit away. And it has plenty of mitochondria to supply it with all of the ATP that it needs for fusion and then for recycling vesicles. Talking of recycling, well, you can't just keep on fusing vesicles into the plasma membrane. If you do so, at some point, your nerve terminal just swells up, and you run out of vesicles. So what you actually do is, well, I didn't do that. What happens is you fuse to the plasma membrane, and then there are many mechanisms which will take the vesicle back and put it back into the vesicle cluster. One of the most famous, clathrin-mediated endocytosis. You have a clathrin coat forming onto your vesicle, and essentially ends up with a clathrin-coated vesicle, which is thought to be almost perfectly identical with the one that fused, and then this loses the coat and forms a new vesicle. And Susanne also told you that I was in Bill Batz's lab. In his lab, what I learned was that one of the most fun things to do with the synapse is to put a fluorescent dye outside of the synapse, take your preparation, have it in a water bath, put a fluorescent dye outside of the synapse, and then as the vesicles fuse and then are reformed and go back from the plasma membrane inside the vesicle cluster, they will take up the fluorescent dye with them. So that's not really a big deal. I mean, it allows you to look specifically at all of the recycling, the active synaptic vesicles. And you can do this with all sorts of stimulation paradigms. You can do it with only a few action potentials, and then you have only a couple of vesicles labeled. You can look for how many vesicles are labeled with very strong stimuli. It's plenty of stuff to do with such a technique. The problem is, resolution doesn't really allow you to look at this one vesicle and recognize it as such. So for example, if just this one vesicle here is labeled, the diameter of it is approximately 40 nanometers. Diffraction limit says that it's going to look like a blob of about this size. So whose fault is that? And it turns out the fault for that belongs to this gentleman over here. This gentleman is Ernst Abbe. He lived in the 19th century, and he worked with a very well-known gentleman called Carl Zeiss. Does anybody know what Carl Zeiss became? He became a company who now sells microscopes, pretty good microscopes. Ernst Abbe did not become a company, or rather did not found a company. What he did, he founded theories on how actually how light goes with lenses and mirrors. So his theory of diffraction of light, for which he is mostly remembered, was developed in the 1870s and published in 1873 when he was working in Carl Zeiss's lab in University of Vienna. So in 1873, so quite a few years ago, what Ernst Abbe proposed was if you have light here and you focus it through a lens to a spot, the size of the spot where you focus your light is not infinitesimally small. Light is a wave. You cannot really keep a wave in a tiny, tiny spot. It will have a physically defined size. And he said, well, the diameter, the XY diameter in the focal plane, will depend on the wavelength of the light you're working with, so on lambda, and on 2 times n, the refractive index, times sinus of alpha, alpha being the incidence angle. And n sin of alpha, anybody knows what n sin of alpha became in the hands of the infamous Carl Zeiss and in any microscope today? It became those funny numbers that are followed by NA on any objective. 2 times n sin of alpha, that's the numerical aperture of the objective. And the numerical aperture gives you an indication on how small or how large your focus spot will be. So what is commonly written after NA on a 100x objective, oil immersion on a Zeiss microscope or Leica microscope, Olympus microscope, any microscope, Nikon, whatever? Anybody knows? Thorsten, you know? 1.4, that's right. So the gentleman in the front row is right. So Thorsten is a good friend of mine, by the way. That's why I'm likely to make another joke with him later on. I will not pick on other people. So if we have a numerical aperture of 1.4, and we are working with lambda, with a wavelength of, let's say, with an argon laser that we use for GFP imaging, that is 488 nanometers. Well, if we have 488 and divide it by 2 times 1.4, well, we are not getting a hell of a lot under 200 nanometers. And this is not equal, this is approximately equal. So basically, with the best imaging, with, let's say, the best argon lasers, the best objectives, the best, let's say, typical oil immersion objectives, we will end up with about 200 nanometers. So remember, the vesicle was 40 nanometers. So every 40 nanometer vesicle will now look like a blob of 200 nanometers. And if we have 3 or 4 together, well, it's going to be each will give a blob, and 3 or 4 blobs together form an even bigger blob. Well, it's not going to look that cool. So I should say this is a law of physics, and it's written in stone. This is what we always like to point out. This is the monument of Ernst Abbe at the University of Vienna. And his law, diameter of the spot is lambda over 2 times numerical aperture, is written in stone. This is not, we didn't engrave that. That's somebody did it probably about 100 years ago. So it took until 1994 for somebody to come up with a concept which went away and went to higher resolution than what this physics law says. I mean, this is a physics law which describes how light works. You can't really break it, let's say. You can only, let's say, you can get around it. And I like to show the original paper. This came from the laboratory of Stefan Hell. Stefan Hell is now working in Göttingen, just about a kilometer from my lab. And I worked with him for quite a few years now. And this is the original paper. I will tell you in a few minutes how exactly STED works and what exactly it's all about. But I want you to notice one thing. It's optics letters. To most people in biology, it doesn't say all that much. To me, I never heard of it. It was at University of Turku in Finland for a very good reason. Stefan Hell is German, but because of his crazy ideas, could never get a job in Germany. So he actually had to go and get a job in I cannot pronounce that name, I'm sorry. I was in Finland a few months ago, and they did teach me how to pronounce this, but I forgot. So, but he was in this city well, actually, no, that's a street, sorry. He was in Turku. That's a street. No, that's a street, that's a zip code. He was in Turku, Finland, okay? So Turku I can pronounce. Turku is not the capital, but it's one of the bigger cities, and it's actually a pretty good university, one should say. But this basically is the only one that gave Stefan a job at the time that he was inventing his stimulated emission depletion technique. What I found remarkable about the paper is that it has only 5 references, out of which the first, the fourth, and the fifth are books. So he just says, well, I invented something new, and you can read in books about everything else, and there are just 2 papers that he actually mentions, one of which is his own previous paper. So before you start laughing about this, it is innovative. There wasn't that much that he could talk about before somehow. Nowadays, there are many other concepts, I should say. Many other techniques came from 1994 up to now, especially since 2006. There are 3-4 concepts that really flourished. But STED still remains the first one. So how does it work? It actually uses a principle which stems from that great guy and smarter guy than pretty much every physicist in the last 100 years, Albert Einstein. Albert Einstein in 1917, so 93 years ago, if I calculate right, he proposed that you can use excitation light to turn on a fluorescent molecule, and then you can use another high-intensity light to turn the same molecule off. So basically, and this works remarkably well if I have 2 color lasers, and I guess there is a red laser somewhere around here. There we go. Yeah, there we go. So the idea is the following. Let's put it over here. So let's say we use this green laser to excite our fluorescent light, and then we can use a red laser which we can put right on top of it, and if our hands wouldn't shake, it would really work, but the hands shake, so it doesn't work all that well. But let's say if it were to work, and well, I got it for about 1 second. Anyway, the idea is pretty simple. You use this laser to turn the dye on, and you use then this laser to turn the dye off. Well, what did you achieve? Not much. You achieved about as much as you would do if you wouldn't start the experiment, and then at least you would save the money for the lasers. You wouldn't need to buy two very expensive lasers to get darkness. Well, so the principle that Einstein invented didn't get much use for the next 80 years approximately. But in 1994, what Stefan Hell proposed was the following. He can use a green beam, just as I showed you, a green excitation beam, focus it through normal objectives, and focus it into one spot on your fluorescent sample, and then quench the fluorescence in this sample, but do it a little smarter than what I said. Quench it with a second laser, which we use a phase modulator to make it look like a donut. So now this one has a minimum intensity in the center, and its maximum intensity is on a toroidal shape, on a donut or bagel or whatever else, on something with a hole in the middle shape. How does it work? It's a fairly simple principle, as Einstein thought it. You turn your dye from the ground state to the excited state with your excitation light, and it can either fluoresce and emit a fluorescence photon, or if you disturb it while it is still in the excited state with a different beam, it will force it to give a photon of the wavelength of this beam. So basically, if I disturb the fluorescent dye with a deep red beam, it will have to emit a deep red photon. And my dichroic here will simply cut that photon out. So what is the net result? Well, if you excite this green bubble and then you quench the edges of the green bubble, the net result is you're only collecting fluorescence from the middle of the green excited spot. So you are collecting fluorescence from a smaller field. You're not breaking the laws emitted by Abbe, but you are getting around them. You are exciting a normal big spot, but you're only collecting fluorescence from the middle of it. Very clever. So how do you get an increase in resolution? Well, fairly simple. If we now just start, let's say we start with a green laser, we just put the green laser here, and then we are going to increase more and more and more the power of the quenching laser. And here, we are going to measure fluorescence coming out. So if we just have the green laser, everything, all the fluorescence we excite comes back to us, we get 100% or 1. If we now start to increase the power on the quenching laser, the donut, the hole inside the donut, is not diffraction limited. What's diffraction limited is the size of the donut. but not the hole inside. That has nothing to do with diffraction. The size of the hole only depends on how much power we put on the laser. So if you buy a more and more expensive laser from various companies which I could name, but they don't pay me to name them, so I won't name them. If you buy a more and more powerful laser, well, the donut actually gets more and more intense, and the zero in the middle gets smaller and smaller and smaller. So eventually, you end up with only a tiny point in the middle of the donut from which you allow fluorescence to come to you. So although you actually excite all of this area here, you're only collecting fluorescence from this inner center. So what exactly does it look like? Well, it looks like that initial picture that I showed you. This is basically a neuron, and I'll tell you in 1 second how we label it. It looks kind of like this. I mean, from confocal, you now start to see individual spots. But now let's just go back. Let's go back 1 second. So, have we understood so far? Are we comfortable? I mean, do we kind of trust this technique at the moment? Or, I mean, he published about 200 papers in the meantime, so I guess, it's probably something we can trust. So let's say we kind of understand we have excitation quenching all around the excited spot. Only the deep center of the excited spot can be measured, so we get better resolution. In this case, for example, with this microscope, if we look at the size of the small spot, it's about 66 nanometers in diameter. And you remember that the original spot was about 200 nanometers in diameter. So the first time I went to Stefan's lab and I looked at this one, I said, oh, so you get about 3 times better, big deal. And he was somewhat displeased at the comment, and his immediate reply was, well, it's 3 times better in X and 3 times better in Y. So it actually is 3 point something times 3 point something times better. So in this case, even though you're from 200, you only go to 66, it actually is about tenfold, roughly about tenfold better in XY, because you're not measuring just in one axis, you're measuring in both. So here you could have 10 little spots and you only see one blob. And if you use this, if you use STED, well, you will see 10 little spots together. But there's a trick. And the trick is not every single dye can accept this type of treatment. What you have to do is you have to have the dye, excite it, and then quench it. And then this is a scanning technique. So you will do it again and again and again and again and again. Not every dye can take this kind of a beating. A lot of dyes will simply bleach out. And a lot of dyes are even more nasty. They get excited instead of quenched by the STED beam. So when you look at the spectrum of, let's say, fluorescein or whatnot, it has a maximum of excitation and a long tail, and that tail looks like nothing. But this beam is very, very powerful. So if you even have a little bit of excitation at very long wavelengths, well, the STED beam, instead of quenching, is going to excite your dye. So all in all, what it ends up with, at the moment, there are a few chemical dyes which work quite well with this technique, and of the GFPs, the only one which is really nice to use is citrine. So we said, okay, we'll just use chemical dyes. They are anyway more stable than citrine. And what we did was then we want to look at synaptic vesicles. And we know synaptic vesicles will eat up pretty much anything you put outside of the prep. So we said, well, we'll just use, we know that synaptotagmin, a vesicle marker, has a domain inside of the synaptic vesicle. So then we said, well, we'll just feed the cells with anti-synaptotagmin antibodies. As the vesicles fuse to the outside, the antibodies go inside the vesicles, and then the antibodies are taken up into the newly formed endocytosed vesicles. And if we label these antibodies with our chemical STED-efficient dyes, then we'll have pretty pictures. And that's precisely what happened. It is a synaptic-like stain. I mean, this is a neuron in culture, very low resolution. This is 10 microns size bar. And every blob here is a synapse. If we now turn on the STED and increase our field size a little bit, from the blobs, we actually start to see single vesicles. This is about 1 micron, so every blob here is probably about 60, 70 nanometers in diameter. Was close to single vesicle resolution, and this we published a while ago already. That was in 2006. We were quite happy with it, and it took about, each picture took about roughly 10 minutes to make. It was, and in those days it was about, what do I know, about 2 by 2 microns take about 10 minutes. Of course, the goal we had was to actually characterize movement of vesicles. So if you take 10 minutes to take a picture, well, that didn't seem particularly achievable in 2006. And again, I went to Stefan and I said, well, if you can only take a picture every 5 minutes, it's not going to be any good. And again, he was rather displeased with the comment, and his reply was better than the first time. His reply was, wait. And it actually only took about 1 year until I had something to wait for, sort of, until we got the first live pictures. So now, how do you get live? Well, you get a little better detection systems. You build a better microscope. But at the same time, you go for a much smaller area. So everything that I'm going to show you from now on is going to be now live movies of preparations. But we're only going to look at areas of about this size. So it's only 1 or 2 boutons in an axon. It's pretty small areas because, of course, a small area you can image faster than a big one. And these were some of our first ever pictures of live, living, moving synaptic vesicles. You might say, well, it ain't moving. It's not. It's just 2 frames. But the movie will come in only about 1 minute of blah, blah from me. So first of all, what does it look like? Well, this is a confocal frame. This is instead a first frame. We are acquiring pictures now at a rate of 28 hertz. So each frame takes approximately 36, 37 milliseconds to acquire. It's very fast. So the intensity that we collect from every vesicle, here the arrowheads show a few vesicles, intensity we collect from every vesicle is not very much. So you can see in 2 consecutive frames, I mean, this is let's say, a time 0, and this is a time 37 milliseconds. Some of the vesicles can be certainly recognized in the same places, and some, like this one, well, that disappeared. So it probably moved up here. Well, something like this is not entirely convincing. So what we thought was, well, let's filter it a little bit, and let's try to enhance signal to noise. And to do it properly, we did exactly the same procedure on the confocal. Well, confocal doesn't look a hell of a lot better by filtering. But if you apply a slightly blurring filter to the STED pictures, you start to get a lot better signals, and your signal to noise looks remarkably better. So again, most of the stuff that I'm going to show in the next movies is going to be filtered, and occasionally we have the raw data playing alongside it, but most of the stuff, it will be disfiltered. What's the resolution? Well, originally about 260 nanometers. Resolution after STED, well, about 60 something nanometers. Not every single vesicle is labeled here. So because they are a little bit sparse, well, this resolution is good enough to really see single vesicle. In single vesicles, certainly the confocal is not good enough for that. So without further blah blah, this is the first movie that we got. This is the raw data. As I said, well, it's not that easy to follow everything, but things are much easier to follow in the slightly blurred but strongly signal to noise enhanced filter. So what do you see? Well, you see spots zooming about, I hope. If not, you're blind. This is basically what we saw originally, and we did this many times because this is not supposed to be like that. I mean, most studies so far using confocal or epifluorescence imaging have suggested that movement is probably not very strong. FRAP experiments, if you bleach one synapse, well, in 10 minutes you have about a 10-20% recovery. So there is movement for sure, but it's not that much. Well, we see an amazing amount of motion, and essentially no vesicle stays put, let's say. Well, there are a few maybe that stay put, but not so many. So what do you do when you find something, let's say, remarkable? Well, you first publish, and then you continue to research on it in an attempt which in America has been called shoot first, ask questions later. So this is basically what we did. We shot first and continued asking questions. So I should say the typical view of how movement should be in the synapse is this one. You have a cluster of vesicles. In principle, they don't really do anything. When you stimulate, some vesicles move and fuse to the plasma membrane. Then they might diffuse into the plane of the membrane. And afterwards get endocytosed and actively moved on top of the vesicle cluster, where again they will not do anything. And every once in a while a vesicle might exchange from one synapse to the next one. Overall, there just shouldn't be that much motion. We didn't really see that. We saw a much stronger amount of motion than a typical model would tell you. So we said, okay, well, we'll just continue to look at it. This is now another movie. This loops over and over through a comparison between confocal, here filtered and here raw data, and then STED, again filtered here and raw data here. It's not that the STED makes a move. I mean, STED allows you to really see nice movement, but there is some movement detectable in confocal as well. So at least the movement that we observe is not a STED-induced artifact. You can also try to look where the movement takes place. So in this case, we have an axon labeled with the mitochondria inside labeled with mitotracker, and you see a little mitochondrion right on the side of the axon. Well, as you will see, this is, I always complain to my student that this is very dim and I don't see anything in it, but I think he does see something in it. But, well, I don't, so maybe let's concentrate on the filtered image. Well, if you look at the overlay, which is filtered and mitochondria, you can see that most movement takes place into what really looks like a synaptic bouton alongside the axon. And every once in a while, you see something just zooming and going pretty fast in the axon. So it does look right. I mean, they do seem to move in boutons. And when we quantified movement inside the bouton, it is a lot slower than in the axon. So it does seem reasonable, but there is a lot more movement than we should have expected from the literature. So the first conclusion we came up with was synaptic vesicles are highly mobile. And without further waiting, we published this conclusion. The whole of the experiments for this publication, I should say, took the remarkably long time in Stefan Helsey's lab of 2 weeks. Normally, it should be 1 week, I was told. Apparently, I was just not very efficient. So, but then we said, well, let's spend now 2 years and try to figure out what's going on. And this is exactly what we did. And one of the first things that happened was, I'm going to put the hand over here, and I'm going to play this movie a couple of times. This is now the only full-length movie. This is exactly what we see when we do an experiment. We take a movie for about 37 seconds or so. We basically image until the computer card is full, and then we stop and we transfer the images. And what you will see when it will start looping again is that there is something pretty magical happening in this area right above the hand. So the movie will start in a few seconds, and you will see at the beginning lots and lots. Well, it doesn't start because it doesn't loop. What do you know? At first, there are lots and lots of vesicles around here, and there is one that just kind of keeps stuck over here and stays and stays and stays. And I said, well, big deal. So what? I mean, there is some spot there, maybe it's an active zone, and this vesicle stays docked to it. Well, there is something a little more strange than that. This vesicle can't stay there that long because one vesicle is bleached within 2 seconds. So it can't be the same one. So this bright spot over here was bright for substantially longer than 2 seconds. It's bright probably about half the time the movie runs, so that's probably about 15 seconds or so. While pretty much everything else eventually is gone, and you can appreciate the fact that that bleaching does take place, things do disappear. So we thought, well, that spot is kind of sticky. It can't be just one vesicle because it would have been bleached. It must be that a vesicle goes in there, gets bleached, and another one comes, gets bleached, and so on and so forth. So it got us interested in what we could call sticky spots. So how do you figure out a sticky or hot spot? Well, you take the whole movie, make a sum of it. Whatever comes up in a summed movie will be areas where things most of the time remain stuck. And occasionally we see very small spots. I mean, in this case, this spot is just 74 nanometers in diameter. So it's a really tiny spot where things wanted to stay stuck. Sometimes it's a little more blurry with the spots kind of diffused around in the axon. And occasionally you don't have so many of these hotspots, but rather you have things that look more like canals or something, which we called rather unimaginatively, we called tracks, because it looked like the Straßenbahn or the train tracks. And again, I mean, where would you expect to have more motion? Well, if you're stuck in a spot, you're probably not moving that fast. And if you're on a track, you're probably moving fast. Well, that's exactly what happened. But we also realized that during time, these hotspots kind of remain in one place. So if you take an image of the hotspots and then you bleach every single vesicle away and take later another image of the hotspots, which now has to come from new vesicles, vesicles which afterwards, sort of after the first one was bleached, new vesicles came into the synapse. They came and parked pretty much in the same hotspots. So it's kind of an unescapable conclusion that some vesicles are more or less immobile, at least in some areas. So first conclusion, everything is mobile. Second conclusion, maybe not. Maybe it's not that mobile. At least there are some areas, some sticky areas. And we did want to say, well, those are active zones. There's just one problem, it's too many of them. There are very many, and they are all over the synapse. Knowing the size and number of active zones in this prep, it just doesn't add up. They cannot be active zones. They have to be something else. And then we did another experiment, you see, and that's where all the trouble started. So we said synaptic vesicles are highly mobile, maybe not all the time, but certainly they're mobile. But then something happened, which I like to call the lunch experiment. So what happened was the student always labels vesicles with antibodies. So he puts the antibodies on the cultures, washes them, then puts them on the microscope, starts imaging. Those are recently labeled or recently endocytosed vesicles. And one day, don't remember whether it was a nice day or not, he decided it would be a good time to go to lunch. So he took the preparation, put the antibodies on, labeled the vesicles, washed everything away, and was like, well, now I could start imaging, but I'm hungry. Duty calls, or rather stomach calls, takes this culture without antibody, it's all washed now, ready to image, but just take it, put it in the incubator, go to lunch, come back, have coffee, come back, and then start imaging. Apparently about 2 hours later. And what do you think happened? Well, things changed. This is now an incubated preparation. There is a lot less motion. So if you see the recently endocytosed vesicles, everything is moving about. The incubated preparation looks just like it's bleaching out. And that's exactly what's going on. It's just bleaching out. The amount of motion you see in there is just artifactual motion. It's identical to the amount of motion you see in an aldehyde fixed preparation. So then we can quantify this in various ways. I won't bother you too much with how we did it. But if you focus just on this panel here, these are trace speeds. These are vesicle speeds. And let's just compare no incubation, which is this lower one, with then the incubated in red and the aldehyde fixed one in the dotted line. If you incubate, you are essentially about as mobile as if you were fixed with aldehyde. So something really, really funky happened in the 2-hour lunch period. Not just that the student was satisfied and had a full belly, but the preparations also changed movement behavior completely. So an easy conclusion from this would be that the recently endocytosed preparations are mobile, or rather the vesicles are mobile if they are recently endocytosed, and after a while they get immobile. And a simple experiment was to try to see, well, where do they get immobilized? The most logical assumption would be that they get integrated into vesicle clusters. So we said, well, to check for that, we can either take non-incubated or incubated preparations. We have our red label, this synaptotagmin antibody that we always put on, and then let's fix the preps and let's immunostain them, let's say, for synaptophysin. Synaptophysin is found on all vesicles, including these mobile or immobile ones. But if you're doing a confocal image on them, an 8-bit confocal image, well, you're only going to focus on the big clusters of synaptophysin. Synaptophysin in single vesicles is going to be almost invisible because it's just one vesicle in one place. All you're going to see is big blobs of vesicle clusters. So let's just take the green here, as vesicle cluster staining. So if you stain vesicle clusters, Without incubation, you don't have such a great correlation between synaptotagmin, between your recently labeled vesicles and the vesicle clusters. If you draw a line through them, well, the peaks are not particularly well correlated. If you wait for a while, all of your recently endocytosed vesicles end up in nice clusters. And if you draw a line through this, the correlation increases quite a lot. And we looked at a whole bunch of different proteins. We looked at all sorts of vesicle cluster or even active zone stains. And the overall conclusion is that if we look at our no incubation in gray, our no incubation preparations, these bars on the left, they don't colocalize that well with either vesicle markers or active zone markers because they seem to just be moving around all the time. But if we wait, the correlation, look at the light gray bars, significantly goes up for all of the vesicle markers or the active zone markers. So it tells us our vesicles are initially running around, and then after incubation, they end up in clusters. So I'll jump over what the meaning of this stuff is in the interest of time. A second conclusion is if you wait long enough, all vesicles become immobile. So you see it's more and more kind of explaining the original conclusion that we published. Well, everything is mobile. Well, maybe not, really not. And let's see what else comes. So then the next idea is, well, mobile or immobile. The only thing that vesicles are supposed to do is they are supposed to fuse upon stimulation. When you start stimulating a preparation, they do exocytosis. And most cartoons will tell you that when you stimulate, things move, they go to the active zone, they fuse, and everything gets active. So we said, well, we'll just test that. So we took incubated and no incubated preparations, and I just jumped over one slide, but we'll come back to that. I just find it easier to talk about this one now. We took incubated, no incubated preparations, and we said, as you see in the red stimulation coming up here, we stimulate, what a surprise, at 20 hertz for 2 seconds. And sometimes when there is time, I actually make a poll from the audience who sees a difference and who doesn't see a difference. And as not very many people are typically ready to give an opinion because they're a little shy, I won't make a poll this time. It was not successful the last 3 times. So I will just tell you what it is. Well, there is no change whatsoever. This one doesn't really move much and is just bleaching out. And when you stimulate, really nothing happens. And this one moves like crazy and does bleach out at the same time. And when you stimulate, still moves like crazy, doesn't change at all. And you can analyze this. You can get curves of vesicle speed during stimulation and before stimulation. And there is essentially no difference whether you use no incubated or incubated preparation. If you use the preparations where vesicles are pretty stuck, they remain stuck. If you use the preparation where vesicles are mobile, they remain mobile. And stimulation doesn't cause any change whatsoever. So I'll go back now to the slide that I skipped. And that was, if now we know that there are mobile and immobile vesicles, a simple question would be, can they be exchanged between synapses? And again, the normal hypothesis would be if you have something that's 100% stuck, well, it's not going to move out of the synapse. But if you have something that's truly mobile and runs about, that will exchange much better between synapses. And that was over here. And that is remarkably wrong. So basically, how do we do it? We just do FRAP. This is about the only figure of conventional microscopy in this talk. We just bleach a synapse, and then we just wait for signal to come up in the bleach spot. And we hypothesize that if you have incubated preparations, the ones where vesicles are not that mobile, well, you won't see any recovery because they're not mobile. They're simply not going anywhere. And the ones that are recently endocytosed, they're much more mobile. You should see more recovery. Well, both are the same. In both cases, you see exactly the same, without any significant difference, exactly the same recovery. And the only thing that, fortunately, is actually not recovering is the fixed preparation, the aldehyde fixed. So at least we know one condition where things can't move at all, and that is if you fix them with aldehyde. That's good. So it's very confusing, isn't it? We have immobile and mobile vesicles, and both exchange between synapses. What does that teach us? Well, that nature is complicated and our experiments are not very able to solve it, but it also tells us that the mechanisms of synaptic interchange are different than the basic mobility. So we conclude that confusingly enough, both mobile and immobile vesicles exchange between synapses, and that doesn't have anything to do with them being immobile or highly mobile. It's probably an active mechanism which simply doesn't care whether that's a mobile or immobile vesicle. If you grab a vesicle, then you just send it to the next synapse without really caring on what it actually wanted to do. So now we'll skip the stimulation part, and now we're back here, and basically we know it's getting more and more confusing. We have vesicles, they start out being mobile, they can get immobilized. Whether they are immobile or not, they still exchange between synapses. So the conclusion was then, well, stimulation doesn't change anything. Activity has nothing to do then with the vesicle mobility. Everything is somehow time-dependent, but not activity-dependent. And as every time in this project that we made a hypothesis, it was wrong. So what we did was we did the opposite experiment. We silenced the cultures. So we said, well, if activity has nothing to do with mobility, then if we silence the cultures, we shouldn't see any difference. So we silence the cultures with TTX, which does what now? TTX? Tetrodotoxin comes from the fugu fish, kills a lot of Japanese every year. What else it does? Torsten? It blocks some channels, eh? Sodium channels. Sodium channels, that's right. It blocks sodium channels. You have no action potentials. You have no activity whatsoever. So it's completely the opposite of stimulation. You can't really tell much, and I cannot really follow these things by eye and tell that whether there is a difference. But the histogram actually shows a significant difference. If you silence the cultures, the mobility of the mobile pool of vesicles actually decreases very substantially. So activity does seem to have a role, although stimulation doesn't increase movement. So moderate stimulation doesn't really change mobility, but synaptic activity is still needed for movement. So the last thing that we said, well, we are confused enough. We have just one more step of confusion to go through. We said, we want to see what happens on the plasma membrane. And what we did was we treated the cultures in such a way that when we fuse vesicles, the molecules, the synaptogmin molecules, and hence our antibodies, remain stuck on the plasma membrane. And that is not that easy to do, because every time you stimulate exocytosis, endocytosis just follows. But there are a few tricks. You can use laterotoxin, which is contained in black widow spider venom, or you can use solutions without any divalence. They will block endocytosis. So whatever is on the plasma membrane will stay on the plasma membrane, and then you can monitor movement in the plane of the membrane. And what's good with black widow spider venom, it also increases the amount of vesicles in the plasma membrane, so it even pushes stuff in the plasma membrane. Makes your terminal look a little bit like a ball. We controlled whether the stuff was actually on the plasma membrane. We'll skip over that. So without any further ado, this is what it looks like. If you have stuff in the plasma membrane pushed out by black widow spider venom, your synaptic bouton, from being a small spot or a small area, becomes a little bubble, and stuff moves in the bubble. If you stimulate more mildly the preparation by actually using caffeine, which does a little bit puts a little bit of material in the plasma membrane in absence of any calcium, you have also motion, and in a control condition where you're just looking at what the equilibrium is on the plasma membrane, you also have motion. So what have we learned out of this? Well, there is motion in the plasma membrane at any point, or whether you increase the amount in the plasma membrane by stimulation or not. And to me, it looks like this is a little faster than this one. This looks a little slower. And we actually quantified it. And it's a complicated slide. We'll only look at this part. My PhD supervisor I actually met him last week. He would say, Silvio, if you're not talking about the rest, why do you show it? I forgot about it. I would have showed just this one. But let's ignore the rest. If we now look at these trace speeds in the different plasma membrane stainings, you see the no divalence, which is a control situation, just equilibrium, just whatever is in the plasma membrane at equilibrium, doesn't have so high speeds. It is comparable with vesicles inside. But once you put black widow spiral membrane or caffeine, these two increase the amount of material in the plasma membrane by about 60% or by about 100% or doubles it in case of black widow spiral venom. Both of them end up with substantially higher motion. You have to imagine that fixed preparations would look like this, would just be this peak over here. So actually what we compare is just this part of the graphs. So what does that tell? Well, there is motion in the plasma membrane. At rest, this motion is not that high. You can increase it by putting more material in the plasma membrane. But that material is not infinitely, let's say, zooming about. It moves at a fairly slow speed, about as fast as a vesicle inside the terminal. So, to conclude from this one, well, movement on the plasma membrane can be traced. It's relatively restricted. I honestly expected that things would just diffuse out and just zoom about, and we would never be able to follow anything. So overall, I know that this is highly confusing, and it's highly confusing to us as well. I just wanted to give you an idea of what kind of stuff we can learn with new technology. I will not say that we got a model which is a lot better than the original model. The original model said, well, we have a cluster and things fuse and come back to the cluster and that's it. Well, through all of this high-resolution imaging, all we managed to do is just to make things a hell of a lot more complicated. So what did we learn? Well, we have a cluster of vesicles, shown here in blue, mature, let's say, or old vesicles, which are immobile. Once you have them fuse, they become recently endocytosed, or rather, recently exocytosed would be vesicles in red. Some of these things are stable in the plasma membrane, and some are mobile, but it's kind of limited mobility. It's not huge mobility. And after fusion, they get endocytosed and now become recently endocytosed vesicles, which, for reasons which only they know, are very mobile. And then these ones lose their mobility over a few hours, and that's activity dependent. The more activity you have, probably the more time this just comes back down and fuses and goes through another round of being mobile. If you stop them from coming down and fusing, eventually they lose their mobility and integrate into the synaptic vesicle cluster. And confusingly enough, both mobile and immobile ones are exchanged between synapses. So I bet you'll ask me, like any of the last few people that saw this talk, you'll ask me what the difference is between the recently endocytosed and mature vesicles. And I'll tell you, I have no clue whatsoever. I mean, this is just speculation. But I have remembered recently something that I read a long time ago, and that is synapsin doesn't bind to fused vesicles. So now, what is synapsin? Synapsin is a molecule which constitutes the glue which holds vesicles together in the cluster. If you take synapsin away, the clustered vesicles seem to disperse. So what happens if synapsin doesn't bind to fused vesicles? Well, if you have a vesicle, it just fused, well, it lost all of its synapsin. When it's being put back, it doesn't have any synapsin on it. If it doesn't have any of the glue that sticks it to other vesicles, well, it will stay mobile. That's a simple interpretation. If it fuses again, so much the better. Again, it doesn't have any synapsin on it and continues to stay mobile. And if you stop it from fusing, well, eventually synapsin grabs hold of it and eventually brings it and makes it blue, makes it cluster integrated. I would say, the good definition for what I just said is called hand waving, but that's about the only explanation that we have at the moment to explain any of these otherwise rather confusing phenotypes. So that's about it, I would say, and I would just like to thank pretty much the one guy who did most of this work. He's kind of shy, so he's behind here. His name is Dirk Kamin, and he's done all of this work on his own with two wonderful microscopists in the lab of Stefan Hell, Volker Westphal, and Marcel Lauterbach. And Marcel I should say Dr. Marcel, because he just got his doctorate about a month ago. And yeah, then I should like to thank you for putting up with all of this mambo-jumbo for about an hour. Thank you.